Let's start second international online conference, Microplastics and Nanoparticles, Emerging Threat to Environmental Health. And today we have project results which will be presented, and this project has received funding from Research Council of Lithuania, but some delays of main presentation will be because our project head is on the way in Paris, and he will connect. Also, I am from abroad, and I have a weak connection of internet, but I hope that everything will be okay. And we want to make slightly changes in program, and the first presentation will be later when Associate Professor Sajed Abbas will connect to our conference. This conference from presentation which will be done by Emilija Pečiukaitytė. Emilia, are you here? I saw that you are online, and
yes, I'm here. Everything's
yes.
okay.
Can you present your presentation?
Of course.
Because I know you have limited time, and you ask in the morning. So Emilija Pečiukaitytė present her results, which is connected with cancer therapy. And some nanoparticles are included in this research. Emilija Pečiukaitytė from National Cancer Institute of Lithuania. And we have also cooperation with this institute. And together we have also some projects. So Emilija, we ask you to present.
Okay. So, thank you for introducing me. So today I will introduce you to our group's research on near-infrared excitation-based nanocomplex for enhanced photodynamic cancer therapy. So firstly, I would like to briefly introduce what that photodynamic therapy, or PDT, is. So PDT is a minimally invasive cancer treatment in which photosensitizers are activated by light of a specific wavelength. And once activated, these molecules generate reactive oxygen species, which cause cellular damage and ultimately cell death. So one of widely used and clinically approved photosensitizers is chlorine E6, or C6, later on in this presentation. It absorbs light in the UV and visible regions. And however, chlorine E6 has two major limitations. So first one, it undergoes rapid oxidation and degradation when activated, a process known as photobleaching. Second one is that the light which excites chlorine E6 does not penetrate deeply into biological tissues. So together these limits just limit these issues, its effectiveness in treating deep-lying tumors. So to address these drawbacks, scientists have proposed of using up-converting nanoparticles, or UCNPs later on, as energy donors. So when chlorine E6 is loaded onto the nanoparticle surface, typically through a phospholipid or polymer coating, the excitation wavelength can be shifted from red to near-infrared light. And near-infrared light penetrates tissues more efficiently, and potentially it could enable deeper PDT treatments. So in this study, upconverting nanoparticles were synthesized by Dr. Vaidas Klimkevičius and PhD student Eglė Žirskytė. The nanoparticles consisted of sodium gadolinium fluoride doped with erbium, ytterbium, and neodymium ions. And this combination of lanthanides allows excitation with both 808 and 980 nanometers laser light, providing flexibility and reduced risks of photothermal effects. And for surface modification, we used phospholipids, phosphatidylcholine, and polyethylene glycol, or PEG, conjugated phosphoethylamine. So these phospholipids were combined in 3 different molar ratios, and chlorine E6 served as the photosensitizer. More about the synthesis you can find in, about nanoparticle synthesis, you can find in the, if you scan the QR code. So, upconverting nanoparticles were first coated with phospholipids through film formation, and chlorine E6 then was loaded into the coating via passive diffusion. We used 2 human colon cancer cell lines for biological evaluation. And to assess therapeutic response, a live dead staining assay was performed. And during the whole study, we used 2 different sources of light. So 808 and 980 nanometers laser light. So now moving on to results. So basically in the study, we investigated 3 phospholipid coatings with different PEG-grafted lipid densities. And according to theory and literature, high polyethylene glycol content should yield in higher and larger hydrodynamic sizes. However, after surface modification, the opposite trend was observed. And coatings with polyethylene glycol produce smaller hydrodynamic diameters and more negative zeta potentials, indicating good colloidal stability, while coatings without PEG resulted in particles that were approximately 4 times larger, with less negative zeta potential, and there was clear signs of agglomeration and sedimentation. So for this reason, only PEG-containing coatings were selected for further study and experiments. So since chlorine E6 is amphiphilic, the phospholipid coating should act as a nanopocket, a little pocket that stabilizes the chlorine E6 molecules on the nanoparticle surface. So the main question we wanted to answer was whether the non-radiative energy transfer was happening, specifically luminescence resonance energy transfer, if that occurs within this nanocomplex. So after spectral analysis, it revealed decreased upconverting nanoparticles emission intensity in both the UV regions, in UV and red regions, which overlap with chlorine E6 absorbance spectra. So this only indicates, but we cannot completely be sure that energy transfer is happening. So we perform time-resolved luminescence measurements, which are considered to be a definite evidence. So while the average decay time of upconverting nanoparticles alone was approximately 215 microseconds, it decreased to around 100 microseconds upon nanocomplex formation. And importantly, PEG density did not significantly influence this result, and both formulations showed approximately 53% energy transfer efficiency. So while spectral analysis confirmed complex formation and energy transfers, the production of singlet oxygen upon, with near-infrared light irradiation indicates whether the system has a therapeutic potential and is actually working. So by using 808 nanometers and 980 nanometer lasers to excite upconverting nanoparticles, and 405 nanometers laser for direct chlorine excitation, we found that both nanocomplexes generated singlet oxygen efficiently under near-infrared light irradiation, where neither UCNPs alone or chlorine E6 alone produced detectable singlet oxygen. PEG density had only minor influence, with the coating containing 33% PEG, which performed a bit better. Notably, singlet oxygen production under 808 nanometers irradiation was more than 3 times higher while comparing it to 980 nanometers laser excitation. And additionally, a very important part is that chlorine E6 within the nanocomplex showed minimal to no photobleaching, whereas free chlorine E6 was rapidly bleached out under the same radiation conditions, which shows chlorine E6 stabilization and that we could use this nanocomplex repeatedly. And further on, to determine whether the nanocomplex can be used in cancer therapy, we first examined cellular uptake. And after 24 hours of incubation with 2 human colon cancer cell lines, the complex was internalized and localized predominantly in the cytoplasm. And the overlap that you can see in the image between upconverting nanoparticles and chlorine E6 fluorescence only confirms that the complex remains intact and stable inside the cells. Flow cytometry in picture B show that HTC 116 cells accumulated the complex 1.2 times more efficiently than the LD1 cells, and importantly, dark cytotoxicity was really minimal, and after 24 hours of incubation with varying concentrations, cell viability remained above 98%, demonstrating good biocompatibility. Next on, we assessed that PDT effectiveness that I mentioned in the introduction. And cancer cells were incubated with nanocomplex for 24 hours and irradiated with 980 nanometers laser. And after 3 minutes of irradiation, HTC cells viability decreased to 65%, while DLD1 cells were more resistant, showing 91% of viability. So to further investigate dose dependence, an additional 3-minute irradiation was applied to DLD cells, and the viability then decreased further on to 66%, with accompanied by clear morphological signs of apoptosis or necrosis, such as cell rounding and loss of adhesion. Because DLD1 cells are more resistant and 808 nanometers excitation avoids photothermal effects, an extended 808 nanometers irradiation was also tested. And as we can see, viability decreased to 66% after 6 minutes, and even further on to 47% after 15 minutes of radiation, again with pronounced morphological cell changes. So to conclude these results in one place, we successfully developed an upconverting nanoparticle chlorine E6 nanocomplex, which is stabilized with phospholipids and resistant to photobleaching. So the system shows efficient energy transfer, produces singlet oxygen under near-infrared light irradiation, and accumulates in cancer cells while maintaining excellent biocompatibility. And most importantly, it induces significant photodynamic effects, especially under 808 nanometers laser light excitation. So overall, this work highlights the potential of this nanocomplex for deep tissue and near-infrared activated photodynamic therapy. So this is the team. I'm really grateful for working with them all. And if you have any questions or I said something unclearly, I am open to it, so you can just ask. So that would be it from me.
Thank you, Emilia, for a nice presentation. And as Emilia asked you, maybe you have some question because nanoparticles are important, this mechanism, and also for therapeutical purposes. And do you have some questions from audience, listeners? If no, so Emilia, thank you for your presentation. And next presentation we will have from Turkey. Professor Neval Baykan will have presentation about microplastic pollution and risk evaluation in the river basins.
Yeah, hello, good morning.
Hello,
Can you hear me?
hello. Yes, it is our first time meeting, but we know some colleagues from Ege
Oh,
University, and
yeah.
we are happy that you agree to present your results, and I think it would be useful for future cooperation.
Oh, thank you. I hope so.
Okay, and it would be very interesting to listen.
So, good morning, everyone, and thank you for participation here today. My presentation is titled Microplastic Pollution and Risk Evaluation in River Basin. This research was carried out in collaboration with colleagues from Dokuzeli University, Ege University, İzmir Institute of Technology, and Recep Tayyip Erdoğan University. Here is an outline of my presentation. I will begin by introducing microplastic pollution and its effects in globally. Then I will describe the study area, followed by the materials and methods that we use. After that, I will present the results, the ecological risk assessment, and finally the main conclusion of our study. So plastic pollution is recognized as a major global environmental challenge, as you know, and plastic usage increasing every year, and it treats all animals and also the human health also. So plastic pollution especially caused by microplastics, has ecological and health risk in several ways. When consumed by microorganisms and aquatic animals, for example, they can block digestion and affect growth also. And survival of the animals also can affected by microplastics. And also microplastics act as carriers of toxic chemicals such as heavy metals and pesticides, for examples. And eventually these particles enter our bodies through seafood, water, and even air, where may cause inflammation and hormonal disruptions also. The aim of this study is to evaluate the risks posed by microplastics in the Nifsi stream and Gediz River, which are located near Izmir. Izmir is my city, which I lived here for a long time, and our university is located also in Izmir. So you can see some pictures about our sampling areas. Our methodology involved several steps, including sampling, separation, extraction, and finally imaging and identification of microplastics. Each stage was carefully designed to ensure that the results were reliable and reproducible. By combining this approach, we are able to classify microplastics according to size, shapes, and color, and also type. And quality assurance was a crucial aspect of this study. That's why we have some precautions against contamination of microplastics from other environmental media. And here we can see the seasonal, sorry, I forgot to say that we collect 16 river samples and 3 sediment samples from the sites, from the Gediz River and Nif stream also. So you can see the microplastics counts results in both river water and sediment samples. So the results demonstrate that the abundance of microplastics fluctuates throughout the year. Especially in the spring times, we have winter and spring times, let's say, we have more plastics particles than the other seasons. Maybe because of the rainy days affects the microplastic contamination movements or transferring from the atmosphere to the water, for example, or the railway to highway to water media or river water. So here we observe the seasonal variation in microplastics types. Fibers is the dominant one in all seasons, but fragments showed noticeable increases in summer and autumns especially. The seasonal pattern suggests that agricultural and domestic activities may intensify microplastics inputs during warmer months. The differences between river water and sediment samples highlight how certain types of microplastics tend to settle more, while others remain suspended in water. So here you can see the microplastics types. So polypropylene is the most dominant one in the river water, but polyethylene microplastics is in the sediments we can recognize, especially in the winter and also in the springtime. They are much more higher amounts when compared to the others. So, although many of the microplastics we detected were very small, their impact on ecosystem is really large. Small particles can penetrate also biological tissues, so they affect the animals or other living beings. Therefore, conducting a detailed ecological risk assessment is essential to quantify these hidden threats. Risk indices allows us to measure pollution levels and understand their potential impacts on ecosystem and also human health. For the ecological risk assessment, we calculated different indices. These include the pollution load index, the polymer hazard index, the microplastic pollution index, the potential ecological risk index, and the coefficient of microplastics impacts. Each index provides a different perspective from overall pollution level to polymer type hazards and ecological risk potential also. So by using multiple indices together, we aim to achieve a more comprehensive assessment of risk in the study area. So these slides present the pollution load index results for river samples across different seasons. The highest value was observed in spring, so indicating a high pollution level, while winter, summer, and autumn show only minor risks categories. So these seasonal differences suggest that runoff during spring caused by rainfall and agricultural activity may significantly increase microplastics input into the river. So that's why we have the high risk categories in the spring time. So this slide shows the pollution load index for sediment samples. Unlike the river results, sediment values were extremely high in all seasons. According to classification, this corresponds to category 4. It means extreme danger. So this clearly indicates that sediments act as a major sink for microplastics, concentrating pollution levels far beyond those observed in water column. Another indices is the polymer hazard index. So according to polymers that we observed in our samples, we calculated polymer hazard index. So in all seasons, we have the effect of polymers because of the polymers, but in the winter and the autumn, we have the high impact when compared to the other seasons, according to polymer types. And also in the sediments, we calculated the polymer hazard index also. The polymer hazard index values in some stations recorded as medium level, but we can say that sediments has the high impact also according to the polymer types. So this underlines the fact that sediments not only accumulate large amounts of microplastics, but also retain hazardous polymer types, making them hotspots of ecological concern. So the potential ecological risk index combines contamination factors with toxicity response factors. So in river samples, seasonal variations show that risk levels were generally moderate to high, with some peaks reaching dangerous thresholds also. These indices highlights that even when concentration appears relatively low, the toxicity of specific polymers that can be obtained has significantly elevated ecological risks. So for the sediment samples presented most alarming findings in terms of ecological risks. So several seasonal values exceeded thresholds classified as dangerous or even very dangerous regions. This is for the sediment. So this slide illustrates the coefficient of microplastics impact for river samples. Fragments show the highest risk contribution for the river samples, and fragments often reaching the maximum category with values above 0.5. Fibers were also significant, but generally remain in the average levels. In the sediments, the risk associated with microplastics shapes were even more striking because fragments again dominated, with some values exceeded 0.8, reaching the extreme risk category. So fibers contributed to average risk levels, where foams and pellets remained minimal. This finding is important because benthic organisms are directly exposed to this sediment, and the prevalence of fragments poses several ecological threats also. Sorry. Finally, we calculated the risk quotient approach compares the measured environmental concentration of microplastics with the predicted no effect concentrations. So above 1 ratio of 1 means that has the high risk can be seen in this media. So that's why, especially in all sample stations, we have the higher risk values. As a conclusion, we can say that sediments are long-term reservoirs of microplastics pollution, and fragments are the most hazardous type of microplastics also. And ecological risk indices show extreme danger levels. These findings highlight the urgent need for stricter waste management, improved wastewater treatment, and continuous monitoring to protect aquatic ecosystem and human health are necessary as soon as possible, let's say. And here you can see are the main references. So I would like to acknowledge the key references that supported of this study. Here you can see our recent publications about microplastics pollution in this region. So if you are interested in, you can check them all. And before closing, I would like to thank my colleagues and collaborators for their valuable contributions. Without their support, this study would not have been possible. And I would like to thank the Turkish Scientific and Technological Research Council of Turkey for the financial supports. And thank you for all listening this presentation. And if you have any question, please don't hesitate. Thank you very much.
Thank you very much, Professor Neval Baykan. It is really interesting and useful presentation. Maybe someone can ask questions, or we saw that you present also your contacts, and we can directly connect with you by emails also.
Of course. Mm-hmm.
Okay. Okay, thank you. Have a good trip.
Thank you so
And
much.
yes, and the next presentation is also from Turkey. Our colleagues from Ege University will present some data about polycyclic aromatic hydrocarbons, and Professor Fusun Pelit is preparing now. I see a presentation, and we can listen you, Fusun.
Yes, Professor. Hello.
Hello, how are you?
Florida. Nice to meet you again. From
Again.
Poland, we meet before. Also, I would like to thank Professor Neval Baycan for this wonderful speech because she is going to another city now. Thank you very much. I would like to thank to you, Professor Reda, for giving me an opportunity to present this high-level congress. My topic is about the analytical methodology for monitoring and distribution pattern analysis of polycyclic aromatic hydrocarbons in river basins based on chemometrics. With the increase in urbanization and industrialization, the environmental quality of river basins, which serve as a crucial source for irrigation for agricultural activities, has been deteriorating progressively. Thus, monitoring persistent toxic substances in urban water resource is mandatory for maintaining ecological stability and protecting the human health. Among these organic pollutants, polycyclic aromatic hydrocarbons are the most harmful contaminants. Specifically, the polyaromatic hydrocarbons have been found to contribute to the decline of aquatic life and pose a risk to human and environmental health. and they have the capacity to enter water through a variety of pathways, including overland flow, wastewater discharge, and atmospheric deposition. Studies have identified over 400 polyaromatic hydrocarbon derivatives, and some of these have been listed as priority pollutants by the United States Environmental Pollutant Agency and other agencies also. The presence of these compounds at low concentrations in environmental samples necessitates the use of appropriate analytical methods for monitoring the measurement. In recent years, as an alternative to the standard methods used, efforts have been directed towards the development of green sample preparation methods that involve minimal chemical consumption and low waste generation. So for this purpose, the green techniques are used to reduction and proper management of waste, reduction of the number of samples, or reduction or replacement of toxic regions, and minimization of energy consumption. So these micro-extraction techniques are solvent-based and sorbent-based techniques, including the single drop micro-extraction, the dispersive liquid micro-extraction, and hollow fiber liquid phase micro-extraction. In our study, we aimed to include this dispersive method in conjunction with polyaromatic hydrocarbon analysis to assess the distribution of organic pollutants in river water. And these samples collected at different sites within the Gediz Basin in the Izmir region. A green, rapid, cost-effective, and straightforward method was developed and validated by integrating in this dispersive liquid micro-extraction procedure. After sampling these, we collect the different samples from the Gediz River samples. This Gediz River is one of the major fluvial systems in Western Anatolia, with a drainage basin covering 20,000 square kilometers. In this research, water samples from this area were collected at 21 sampling locations across all over the seasons. In the experimental part, we use this dispersive method. We filtered the samples and transferred them into glass centrifuge tube. The dispersive solvent, acetone, and extraction solvent indecanol was added, and the resulting clotted solution was ultrasonicated, and after centrifugation step, the indecanol phase of the emulsion is collected as a thin film on the top of the solution. And then when we incubate this solution to 20 degrees Celsius minus 20 degrees for 10 minutes, it was separated from the undecanol phase, and at the room temperature, we inject these samples to the GC systems. Selective analysis could be performed for each analyte at different retention times. And also in this study, we include the plaquette Burman design and Buxbanken design for optimizing the experimental conditions. The selected factors and coded levels for each polyaromatic hydrocarbons were seen in this table. As can be seen from the Pareto charts for each analyte, 3 parameters were stand out, including the solvent type, pH, and stirring speed. All other parameters were studied under constant conditions. In the next step, we also applied a three-factor Box-Banken methodology to optimize these three parameters. This figure shows the plots of the contour responses corresponding to typical response surfaces obtained under optimal conditions for each extraction. After evaluating all the given data, the optimized results according to the Box-Banken were determined as extraction solvent volume 50 microliters, extraction solvent type undecanol, and freezing time of 10 minutes. And we also make, after this operation, the calibration graphs. As can be seen from the graphs, under optimum conditions, linear responses were obtained for three polyaromatic hydrocarbon species for river water samples. And these are the analytical merits of the method. Seasonally detected polyaromatic hydrocarbons can be seen in this cohort diagram. And the most frequently was the polyaromatic hydrocarbons were detected in the fall, and at least frequently were found in the spring, as an opposite to Professor Neval results for microplastics. As illustrated, and we can see the bar diagram of each polyaromatic hydrocarbon species obtained at each point according to the results of 4 seasons. As illustrated in these graphs, the benzoanthracene and fluorantane were identified as the dominant polyaromatic species during the winter season. And during the spring season, the fluorine concentrations were notably high, for example, at stations 5, 6, 14, and 20, representing locations affected by domestic and industrial wastewater inputs. And additionally, the presence of fluorine and benzoanthracene was observed at stations 2, 7, and 12. And these are the common characteristics of being located under highway bridges. And if we can look at this summer period, the benzopyrene was identified as the predominant polyaromatic hydrocarbon. Unlike other seasons, pyrene concentrations were notably higher in the summer, which corresponds to the domestic wastewater discharge, while elevated anthracene levels were observed at the station 20, also the İzmir Atatürk organized industrial zone discharge including. And when we compare with other seasons, the polyaromatic hydrocarbon concentrations were recorded in the fall with naphthalene, phenanthrene, and benzopyrene. And these are identified as the predominant species. And these are the discharges from also the same from the industrial wastewater treatment plants. Also, we evaluate the chemometrics method, the multivariate analysis of the samples in different seasons. This is the PCA score plots of all samplings. This plot shows that almost all of the winter samples are located in the upper part of the graph, whereas all the fall samples are on the lower part, indicating that the winter samples are characterized by positive PC2 scores and the fall samples with negative scores. And as from this PCA graph, and as the winter and fall samples are relatively well separated from each other, from the seasons data, all seasons data, a second scenario containing only winter and fall samples was also studied in this project. And these are the PCA scores and loading plots of the calculations. The hierarchical clustering dendrograms of samples and variables also are given. And as can be seen, very clear separation of winter and fall samples are obtained. And after performing this PCA and hierarchical clustering analysis modeling of the samples, we include partial least squares discriminant analysis in order to investigate the possibility of the better classification. And as shown in the figure, both 2- and 3-dimensional graphs, very good separation of winter samples from each other, resulting approximately 100% correct classification, not only for the training data, but also for the independent validations that we obtained these values. And high levels of pollutants, including crescent, fluorantane, anthracene, and benzoanthracene were detected at some stations. And in sample, for example, 16 location, the concentrations of fluorantane, anthracene, pyrene, and crescent were much higher than in samples of 14 and 17. And these polyaromatic hydrocarbons are commonly used in the asphalt, plastic, and paint industries. And they are located near the highway bridges also. And in the fall period, we can, if we look in the fall period, the 4 and 20 stations appear different from others. These samples come from industrial wastewater treatment plant effluents, and they contain high levels of phenanthrene and benzopyrene, which are typically used in the paper and paint industries. Also, we evaluate the greenness of the proposed technique. The green evaluation score was calculated by a complex modified GAPI and budget metrics. And the complex modified GAPI score of 76 reveals that the developed method possesses high greenness and environmental compatibility, demonstrating efficient resource utilization with minimal environmental impact. Additionally, the obtained budget value 70 indicates that the proposed miniaturized extraction-based multi-element analytical method demonstrates good greenness and moderate environmental sustainability. And as a conclude, a fast, green, economical, and sensitive analytical method was developed. Chemometric analyses have proven to be highly effective tools for assessing the polyaromatic hydrocarbon distribution patterns in river water samples and improving technical decision making. In this context, the results may contribute to design of more comprehensive and efficient strategies to combat water pollution. Improving water quality monitoring and management softwares also can be considered as an important step towards enhancing the environmental sustainability. And in the end, I want to say that the water resources belong to all of us. And thank you for your attention. We must protect our seawater. Thank you.
Thank you very much, Fusun,
Yes.
for a detailed presentation. And maybe we have question for Fusun, Professor Fusun Pellet.
Yes, I am turning off my camera because my internet is slow. Thank you for your understanding.
Okay. I am also turn off my camera because I have really bad connection, and sometimes they are logging out from this meeting. Okay, Fusun, and we have the next presentation of Ilknur I'm not sure about pronunciation your name, but I want just to say some words that Ilknur started one project with Klaipėda University, and she is now involved in this project only a few days, but I think that her experience will be very useful for collaboration and project. So, Ilknur,
Thank
can
you,
you say
Professor.
yes? Thank you, and
I
can you say some words about you? Because now we have a project together.
Yes. Hello, everyone. My name is MICROPLASTIcs. Today, I would like to present our study titled Development of Novel Limit Extraction Method for Bisphenol A Determination in Water Samples. This work focused on developing an environmentally friendly extraction method using deep effective solvents. Bisphenol A commonly is a widely industrial chemical found in plastics, resins, packaging materials, and various household products. Chemically, BPA contains two phenol groups and forms a colorless solid that dissolves easily in organic solvents, but only slightly in water. Due to its structures, BPA can get into water source and accumulate in ecosystems. Because of its endocrine stimulant effects, BPA can interfere with hormone regulation, metabolism, and human development. Therefore, monitoring BPA levels in environmental water samples is essential. And BPA is commonly used packaging, coating, containers, and even in medical and industrial materials. It acts like an epoxy device helping seal food products. However, its endocrine disruptive properties can alter hormone production, transfer, and signaling inside the body. Humans are exposed to BPA through ingestion, skin contact, and environmental pathways because it mimics estrogen BPA can disrupt reproductive health and thyroid function and immune response and metabolic processes. And especially in early childhood, BPA exposure may be linked to behavioral and developmental issues. One important part of our study is the protective solvents. When two or more compounds interact and form strong hydrogen bonds, and their melting point decreases and effective mixture forms, these DES mixtures can remain liquid at near room temperature and are considered a greener alternative to traditional solvents. These DES are environmentally friendly, biodegradable, non-toxic, non-volatile, easy to prepare, and cost-effective. They are also recyclable and stable, making them excellent alternatives to commercial organic solvents. They typically consist of hydrogen bond acceptors, such as choline chloride, and hydrogen bond donors, such as organic acid amines, sugars, or urea. These interacts allow DS maintain their liquid state at low temperature. Based on their composition, these are classified into four main types, involving various combinations of ammonium salts, metal salts, metal hydrates, and hydrogen bond donors. DES have diverse applications: extraction of bioactive compounds, metal recovery, electrochemistry catalysis, biotechnology, drug delivery, polymer synthesis, and environmental analysis. These green properties make them highly attractive for sustainable chemistry. The aim of our study was to develop a green sample and efficient deprotecting solvent-based extraction method for the determination of bisphenol A in water samples collected from 21 stations along the Nif stream and Geddes River. In this study, menthol was selected as the hydrogen bond acceptor, and different different acids were used with the hydrogen bond donors. Various molar ratios were prepared and tested to obtain stable and homogeneous decontact products suitable for bisphenol A extraction. This table shows all synthesis disinfectant solvent combinations. Among them, this DBS2, which contains formic acid and menthol at the 1:1 molar ratio, produced the highest analytical signal for bisphenol A and was selected for further work. To ensure the high sensitivity LC-MS parameters such as ionization mode, voltage, gas pressure, and mobile phase were optimized. The final optimized continues allow for a strong and stable bisphenol A signal. And this slide summarizes the extraction process. First, the water sample is placed into the tube. Then the 100 microliters of menthol-based decontact solvent is added to the sample. Ultrasonication is performed for the 1 hour. Then the concentration steps, and the decontact solvent sample is fed through the membrane filter into the insert. Mini centrifugation is done for 10 minutes, and then the sample taken into a 50 microliter insert was analyzed on the LC-MS system. Then the LC-MS system and the system in the development of BPA analysis method using decontact solvent, 11 parameters could affect the signal. Plaquette forming design. Chemometric screening technique was applied to identify the most influential factors, and the 3 most significant parameters were selected. The study evaluated flowing contraction, ultrasonication time, centrifuge time, sample volume, salt effect, pH, DS volume, mini centrifuge time, menthol, formic acid, and the decontact solvent ratio is the our condition parameters. The statistical evaluation relates the 3 significant factors: decontact solvent ratio, centrifuge time, and centrifuge time. These were selected for further optimization. This one showed standards. This one showed standard effect volume. Decontact solvent volumes and ratio were the 2 strongest contributors to signal intensity. The correlative chart usually confirms the significant parameters affecting bisphenol A extraction. These parameters were chosen for box-Blanken design optimization. And in this step, combinations of 3 significant factors were evaluated. The response surface model helped us understand how these variables interact. And the model was statistically significant. Base volume and base ratio were the most important variables, while the interaction between centrifuge time and decontact solvent volume was also significant. Based on Blanken results, the surface response graphs were plotted and optimal conditions were determined. Centrifuge run was 50 minutes. The desiccant volume was 100 microliter, and this ratio was 2.2. This table shows the optimum conditions for method for optimization studies. And this chromatogram shows our clear and strong bisphenol A signal at the 100 ppb using the optimized decontact solvent liquid mixture extraction method. To the test whether the sampling method prior to linear response calibration solution was prepared with concentration ranging from the 0.5 ppm to 1 ppm. After achieving optimal separation for bisphenol A and calibration curves were plotted, the collaboration study was conducted by taking chromatograms of BPA standard prepared in menthol, methanol, water and deep protective solvents matrix. The resulting calibration is telegraphed in the figure, demonstrate the liner's response was achieved for the VPA under optimal conditions. And the recovery studies were conducted for river water samples. Standard additions curve were created by adding additions at low, medium, and high concentration of the calibration curve. And the resulting area values were compared with theoretical area values. 3 replicates recovery results for BPA and present in the table. The analytical characteristic data from the study are present in the table below. To assess the validity and applicability of the developed deep protective solvents with liquid extraction procedure in real river water sample, recovery studies were conducted at 3 different concentrations. And the results and discussion. BPA in water sample taken from the Nifsi stream along the Gediz River and from the İzmir Organized Industrial Zone Treatment Plant outlets for the 4 periods were analyzed with the method we developed and results in the table were obtained. Seasonal evolution related that bisphenol A concentration were highest during winter, followed by spring, autumn, and summer. This pattern may be related to change in water flow, rainfall, and dilution. And thank you for the attention.
Thank you, Ilknur, for your presentation. I have some technical problem. I also was disconnected, but still I am here. And maybe some question we have. If no, so we will save your time. And thank you again for your team. Your team is very strong and we are collaborating. And also I want to present Monika Shmilovska. Also we have together with Ilknur and Monika project at Klaipėda University. And it is very nice to meet Monika. And Monika at the moment has 2 projects at with Klaipėda University as a postdoctoral student, and you just win new 1 project with Lithuanian Research Council, and now Monika will present about plasticizers and microplastic. So Monika, you can start.
Thank you. So today I will present the current challenges related to the migration of alternative plasticizer in the context of microplastics and their transport in the environment, and as a consequence, the potential exposure of consumers. This topic fits into the growing concern over micro and nanoplastics as emerging environmental and public health risks. So plasticizers are used to improve the flexibility of plastics, but many of them are endocrine active and potentially toxic, especially in reproductive and developmental contexts. The main reason for the search of alternative plasticizers is a regulation that restricts the use of phthalates due to their toxicity. As a result, bio-based plasticizers such as citrates or adipates are being developed. For many alternative plasticizers, toxicological data and regulations are still incomplete, and they are still challenging for researchers. In addition, newer alternatives are still emerging, and we don't know what their physicochemical and toxicological properties will be. What is more, micro and nanoplastics can serve as carriers absorbing alternative plasticizers and transporting these chemicals into food, living organisms, and the environment. In addition, plastic production is constantly increasing, leading to a rise in the amount of microplastics. Microplastics enter food through several major pathways. First, they can be released directly from food contact materials during storage, heating, or mechanical abrasion. This process is particularly relevant for plastic packaging or single-use containers made from polymer-based materials. Second, microplastics are already present in many components of our diet. They have been detected in drinking water, processed food, seafood, and even in beverages such as beer and milk. In addition, airborne microplastics can settle on exposed food during preparation or storage, becoming an unintentional source of contamination. Next pathway is related to agricultural systems. Microplastics accumulate in soils due to the application of polluted sewage sludge, compost, or fertilizers. From there, they can enter crops and subsequently the food chain. Each of these pathways represents not only a source of microplastic contamination, but also a potential route of exposure to plasticizers and other additives that may be absorbed onto the surface of these particles. Packaging materials, particularly polymers with lower structural stability, can undergo degradation when exposed to relevant temperatures, light, or fatty food matrices. This kind of stressors accelerate physical and chemical breakdown of the material, leading to the release of microplastics and, as a consequence, the migration of associated chemical additives such as plasticizers. Studies show that the rate of microplastic release increases significantly under heat, such as during microwaving or prolonged storage at elevated temperatures. Likewise, foods with a high lipid content promote the extraction of additives because many plasticizers and polymer-related compounds are highly lipophilic. Moreover, the extent migration is influenced by the type of polymer used, the thickness and structure of the material, contact area, duration of contact, and the composition of the food itself. Different polymers show different migration profiles. The highest levels of additive migration are typically observed in polyvinyl chloride and polystyrene due to their lower chemical stability and higher affinity for plasticizers. In contrast, materials such as PET and PP are generally more stable and exhibit lower migration rates. However, they are not completely free from releasing additives. Alternative plasticizers exhibit a range of biological effects, including endocrine disruption, impacts on the reproductive system, and potential neurotoxicity. Microplastics can further increase exposure by transporting this plasticizer across biological membranes, effectively acting as a Trojan horse. This mechanism is especially concerning in the case of nanoplastics, which are far more capable of penetrating biological barriers and reaching sensitive tissues. This is an example of our results. We studied concentration of alternative plasticizers in protein supplements and their packaging using GC-MS technique at the final determination step. As shown in the diagrams, the highest concentration were detected for DEHT, nearly 14 nanogram per gram in packaging and up to 6 nanogram per gram in the protein supplements, which may confirm the potential migration of this compound. The presence of DIBA and DHA in the supplements also suggests possible transfer from the packaging material or raw material contamination. what is interesting, a compound with the lowest volatility of all compounds analyzed, trimelitate, TOTM, was found at elevated levels in the packaging of product number 1, while its concentration in the packaging of other products was much lower. Similar profile, similar pattern was observed in protein supplement samples, so elevated concentrations appeared only in product number 1. This may confirm the migration of this compound into the food. To estimate consumer exposure, we assumed a daily portion of 30 grams of supplement and human body weight of 75 kilograms. Based on these assumptions, we calculated daily intake values and compared them with available toxicological thresholds. We compared estimated daily intakes, EDI, with tolerable daily intake, TDI, which was established by EFSA or reported in the literature. In most cases, exposure was well below these limits. In this case, migration can occur via direct contact of the powder with packaging, via aerosols generated during handling, or even via micro and nanoplastics acting as vectors. Stepping into the area of challenges, it is important to highlight several key issues. So first, we lack comprehensive toxicological data for many alternative plasticizers, especially regarding chronic exposure. Second, micro and nanoplastics can act as vectors that may increase both exposure and the bioavailability of these compounds. Another challenge is the absence of harmonized analytical methods that would allow simultaneous determination of plasticizers and microplastics within the same sample. Current regulations are not keeping up with the dynamics of the market and the development of new materials. As for future directions, we need low migration or not migrating plasticizers, as well as polymers designed according to the safer by design concept. Highly promising are emerging analytical techniques that integrate chemical and particle level information, allowing for more comprehensive exposure assessment. And finally, long-term human biomonitoring and boosted regulatory frameworks are essential, covering both plasticizer and their interactions with micro and nanoplastics. This is our team of Kujawian-Pomeranian Science and Technology Center of Professor Jan Czochralski in Toruń, Poland. Our leader is Professor Bogusław Duszewski. We are open to international collaboration. And thank you for your attention.
Thank you, Monica, for your presentation. It is really nice to meet you. And next presentation, I will ask Sajjad Abbasi, and later we will have the same order, which is in program. So from Vilnius and Kaunas scientists will present in the second part. So maybe Sajjad is
hello, everyone.
Online. Yes.
Hello again, and thank you for giving the opportunity and your time. As you can see, the title of my presentation is In and Out Flux of Atmospheric Microplastics to and from the Seawater. If you remember the last time, we talked about a similar topic, but was done in the Curonian Lagoon. And we focus on micro layer, but here is another part of the project that was done in the Persian Gulf and Oman Sea that I want to speak about a little. If you remember, the last time we talked about the microplastics movement or transport in the atmosphere and in and out flux from the ocean. And here a little repeat them again. That you can see here in the figure, we show that microplastics can travel long distance across the continents and also on the ocean and continental places through the air, and sometimes just a few days or few weeks. In contrast, transport by water is much slower and may take months or even years. However, it's important to note that these particles these particles can be repeatedly deposited and resuspended again and again. So we have a very difficult movement and hard dynamic for atmospheric microplastics that we try to understand more better dynamic of microplastics, especially in the atmosphere. So if you remember again, the last time we showed this slide that the paper was published. We had a teamwork project 2 or 3 years ago, I think, to identify the information we need, and as well as the information that is already available. So as you can see here, the blue boxes show that data that exists for 2 years ago, while the orange boxes show that where the information is missing. However, I'm sure that we have all of this information, some of them with more paper and some others with lower paper. So keep in mind that so we have all of this data now. And here the red boxes highlight the goal of our study, which was started 2 years ago, based on this project. To achieve these goals, we need comprehensive sampling. So we collected samples from the coastal sediment, land sediments, seawater, atmospheric deposition using the dust collector that I will speak about, and also we get samples with MWAC sampler, or it's better I say that improved MWAC sampler, and we consider offshore and inshore winds using them, and also air vacuum pump. Our sampling strategy was to collect samples from 3 days at each station. In fact, these 3 days include 1 stormy day, 1 calm day, and 1 moderate day. So, and also on each day we took 2 sets of samples during the day when we have onshore, and at night when the wind was offshore. Here you can see a standard MWAC sampler, and as you can see, the tall is 1 meter, just 1 meter. In fact, this sampler was used for dust movement at first, so we try to improve this MWAC sampler that I speak about in future a little. So in fact, this sampler can trap the microplastics or any particles when they move horizontally. For example, when we have a creep or saltation or suspension movement, the sampler is the best to trap them. But here is a problem for microplastics. As you know, the microplastics are very light, so several studies show that microplastics can be resuspended even with low wind speed. So we increase the height here to 5 meters, and we improve the sampler that you can see here that I bring to figure. So this is a upgraded MWAC sampler along the dust collector that you can see in the middle of the MWAC sampler that we install them. Yeah, just this. If you have a question, I can speak about later. And here is a simple dust collector that we try to get samples or microplastics and any particles when they have a vertical movement. It's really useful for calculating flux, in fact, vertical flux, not horizontal. And here we use a vacuum pump sampler. We try to keep the vacuum rate at 14 liters per minute for getting samples. And also sediment samples was done using a quadrat method. And also if you have a question, I can speak more. Regarding the sampling station, after surveying entire of southern part of the Iran, we selected these two stations, one in the Persian Gulf and another in the Oman Sea. In fact, we made sure these stations were located away from any pollution sources. So we think that these two stations can show the can be good indicator for all of this area. So and here you can see some substation in detail. For example, the right figures is about the Oman Sea and the left is about the Persian Gulf. In the Persian Gulf, there are 4 stations. Water samples are shown with a triangle. The other samples such as sediment, dust collectors, and MWAC, and the others that I mentioned, they showed with circles because all of them are there. So I show all of them in with a circle point. Also at the station A4 in the Persian Gulf and A8 in the Oman Sea that you can see in the figure. In fact, we install or consider this station to consider resuspension because they are far from the coastal area. So in fact, we repeated this type of sampling or this strategy for Oman Sea again. I mean, we did the same procedure for both of the sea. So, but in the Oman Sea, we have 2 more sampling sets. The first one is that the land sediment. You can see here. I don't know, you can see my flash or most or not. But here, the land sediment sampling was done up to 200 meters behind the station, because we want to consider the resuspension of microplastics. When you're far from the coastal area, you can have a dry climate, more dry climate or weather. So we expected to have more resuspension. And the second one that you can see here also, M9, D9, and W9 here. In fact, sampling was done in the middle of the sea, about 15 kilometers far from the shore. In total, more than 1,000 samples were collected and sent to laboratory for digestion and density separation and microplastics identification using the visual method and also microrubbers spectroscopy. Briefly, the results show that the fibers was dominant because of several reasons that I can speak if you want. But the dominant size was microplastics lower than 100 micrometer. In this case, because in this case, you know, when we speak about the shape and size, we have a big limitation and uncertainty. because of methodology. For example, in this case, we try to use Raman more, so we detected microplastics lower than 100 micrometers. So we found a lot of microplastics. When I compare my study with my study, I mean other studies, so the number of microplastics much more here. But you know, recently you can see some published paper that they are talking about the uncertainty. So it's really important recently. So we can talk about later. And the color, I think, is not really important. So here, figures show that the comparison of the proportion of fibers among microplastics, and also in other figures, we compare small fibers among total fibers across samples types. For example, dust, sediment, and air pump samples and other things. So in the dust, sediment, and air pump samples are fiber dominated, while for MWAG and seawater and offshore dust, I mean the middle of the sea, contain more non-fibrous microplastics because we detected smaller particles. So it's really important. And here are plotted microplastics number for MWAG sampler at different height. It's a bit complex, but I try to explain briefly because of lack of time. So generally, data were heterogeneous and significant difference in medium concentration were in fact observed between sampling time, because we got samples in 3 days, if you remember, and also greatest, but the greatest concentration was a stormy day always, because of positive correlation between wind speed and microplastics number. And when 3 days data were combined for each location, a significant difference was noted between samples near and far from the coastal area. So this clearly shows that the rate of resuspension is much higher on dry land. You know, in the recent modeling, sometimes they don't consider resuspension. It's really important issue that we mentioned in our paper. And here, the results can be completed when we consider the polymer type. For example, here we can see that lighter microplastics can be more suspended, and they have more residence time in the atmosphere. So they can be trapped by air pump and also MWAG sampler. As you know, MWAG sampler works when we have a wind, just when we have wind. So the lighter microplastics can be trapped in it. And the denser microplastics were found in the sediment and dust collector. The result is logical, because these two type of samplers include deposited particles or microplastics or heavier microplastics. They have a lower residence time, so, and they have more settling velocity, so we have them in the sediment and dust collector. And here we try to calculate settling velocity and residence time and transport distance, but practically, not theoretically. So it's a new things in our paper. Here also we also calculate net environmental settling velocity we try to calculate it for other polymers and different size and shapes. And in fact, and final sentence, say that I can say that this findings suggest that microplastic travels for long distance through the repeated settling and resuspension for several times, and like a grasshopper effect. So it's really important that we should consider in our modeling. And it's better I skip these slides. and we can speak more if you want. Okay, thank you very much for your attention. And finally, I should say thanks for Reda. It's really helped me in all of the project, and Jaron and Andrew that they helped me for doing this project. Thank you very much.
Thank you, Sajed, for your presentation, and also our collaboration will not be finished. Sajed also have a new project with Klaipėda University. He is very active in science, and really we are happy to have you in our team.
Thank you very much.
Yes, thank you, really. And the next presentation will be from Vilnius. Dr. Sergei Shemchuk will present his results, and I want to say, if you have some question, you can contact with presenters, and all participants will get certificates of attendance, and this conference also will be published on site, and we will send all information. Dear Sajed, can you stop sharing, and Sergei Shemchuk will present his presentation, and after Sergei presentation, Sergei will be moderate next presentations.
I will present you my presentation on some studies that I'm doing during my PhD postdoctoral research in Nature Research Center. So I want to present you some results that were got during the studies of the sorption of cobalt ions on hydrogen coated with magnetic nanoparticles in various aquatic environments. And all colleagues that I mentioned here helped for different experiment parts or to characterize the measurements. So I need to mention them also. It's Galina, Danguolė Montvidienė, Vydas Pakštas, Andrius Drabavičius, Martynas Talaikis, Karina Pusborskaja, Kęstutis Mažeika, Jonas Mažeika ir Kęstutis Jokša, bei Živilė Jurgelėnė. It's my supervisor during the project. And what the aim of this study was is to examine the absorption potential of magnetic hydrogen nanocomposite for cobalt removal from different environmental media. So the tasks were to synthesize, then to characterize it, and to investigate the absorption processes. And as you know, the hydrogen, it is the organic biopolymer that is eco-friendly and biodegradable, and it's usually produced from crustaceans wastes, and we can see where it can be also applied just by itself without anything else. It can be used like a sorbent to treat the water, and also it has the two group, amino and OH, so those are always interacting with different toxicants, and this hydrogen also is used in our daily life. We can use it like an additive to a food, and it seems to be very good material to use because it can be easily catched from our life and from crustacean wastes. Also, for a better collection of it, we decided to add him the specific magnetic properties by connecting the magnetite. So then we can collect used sorbent from environment just using the magnet. So first of all, we synthesized the magnetite by dissolving divalent and trivalent iron chlorides, and then with addition of ammonia, the reaction was launched, and together they are combined the magnetite nanoparticles that were grown during the about 2 hours of synthesis, and after they were purified and dried and homogenized for better dosage and to ensure that we can correctly set the ratio of the connection of magnetite to a hydrogen. It's 10, 20, and 30%. So after that, we dispersed magnetite in water and added to the hydrogen dissolved in acetic acid. And after the synthesis, by using the cross-linking agent, the glutaldehyde, we've got the magnetic hydrogen sorbent that just simply was collected or separated from the synthesis solution by magnet and then purified and also dried and used in our experiments. So for characterizations, we used the TEM and SEM methodologies. So we can see here the magnetite on the right. Wait, I'll try maybe to turn on. Oh, I don't know how to make it back. Okay, the magnetite photo, it seems like a dark particles, which are lower than 25 nanometer size, and also synthesized magnetic hydrogen 10%, 20%, and 30% load of magnetite. There are not a huge sum changes, but we can see that in 30% there is more magnetite connected to the hydrogen. The same photos of dried hydrogen and magnetic hydrogen 30% are shown in the right side. So after that, we also recorded the X-ray spectrum pattern that also indicated that we've got a really good connection of magnetite, because all main peaks of the magnetite occurred in all 3 synthesized sorbents in 10, 20, and 30% magnetic hydrogen, and here they were detected by the program that it's really a magnetite and indicated by that score. Also, a FTIR spectra was recorded, and we can see that connection of magnetite was to amino and OH groups. So the main difference that occurred after the synthesis, that the peak at 537 centimeters was wavelength was appeared in hydrogens FTIR spectra. And also we recorded a Mossbauer spectra to ensure that there is no changes during the synthesis of the magnetite, and we can see in the right that there are the similar or almost the similar, maybe some little interactions and difference, but the material has the same condition and it's the same material used. And also we measured the hyperfine distribution field, made a distribution of it, and we ensured that magnetite has a 60 ml per gram strength, and the sorbents which were synthesized as respectively about 20, about 12, and about 8 ml per gram, but it's enough for separation. And then we used different samples from different points like Baltic Sea, Neris River, and Balsis Lake for study our sorbent to collect the heavy metal, it's cobalt, and here are the points where they were taken. And after that, we little bit filtered it through 0.2 micrometer filter to ensure like the fraction that want to interact and to make a big different strong effect on the sorbent, and it won't be collected like some organism or some huge particles from those samples. And after that, we added a known amount of cobalt ions, and after that we added some amount of tracer, cobalt 60, and after different change the experiment parameters like time or concentrations, we separated the sorbent with the use of magnet and after measured it with the gamma spectrometer for detection of the changes in the concentrations. Also in the right, you can see how it works in the sample. It's just enough, less than 30 maybe seconds to ensure a good separation of magnetic higher than 30. So for magnetic higher than 10 and 20, a little bit less. More time required, but still it works very good and can be used in such strategy to collect it from different samples, liquids, and so on. And here are the results. The Baltic Sea samples of our tests, and showed that we have the high enough removal efficiency. For example, for MCN 30 and 20, it's higher than 40. For MCN 10, it's about 25% per hour used concentrations, and also calculated the Langmuir and Froelich isotherms for these results, and also studied the kinetics of the reaction. So we used different contact time from about 5 minutes, from 5 minutes till 1 day, 24 hours. And as we can see, it's enough about 1 hour and 30 minutes to reach quite good equilibrium. And the second slide is in Neris River samples. So here are the removal efficiency a little bit higher. It's about 50 for MCN 20 and 30% at low concentrations, and for MCN 10 it's higher than 30. And the kinetics also, we can see it's enough, again lower than half and 2 hours, half and 1 hour to reach the good results. And also in Belsis Lake, here the most good results for removal efficiency. For MCN 30 it's 70%, for MCN 20 it's about 63%, and MCN 10 showed a 33% removal efficiency. Kinetics are similar, and also we calculated Langmuir and Froelich isotherms. The result of those isotherms are shown here in the table, and we can see, according to the Langmuir isotherm, we've got the best result for MCN 10 in Baltic Sea. It shows like it should be like 36 milligrams per gram of sorbent could be collected from Baltic Sea using this sorbent, and respectively for Neris River it's 994 and Belsis Lake 1239. All in all, analysis of this data showed us that the reaction is favorable, and also that it is hemisorption, monolayered according to the kinetics. And here are the results of all calculation. If somebody needs to look at it and to compare with other research, we can provide it. Also, like in addition to check if it is very good or no, like usually the main heavy metals are used just in simple laboratory studies in pH 6.5 for metals of chromium, nickel, and cuprum. And here, as expected, the sorption capacities are very high because there is no other materials in the sample, like in our used. So the sorption parameters are very high. Removal efficiency can reach even 100 at low concentration. And Langmuir and Froelich and Hill isotherms were calculated, where also we've got quite good results. This one was used just magnetic higher than 30%. So we tried to use the most magnetic sorbent, which is easiest to separate from the samples and to test it. Here are the results, and here are some conclusions that we can say about our magnetic high-to-zone, that it provides high or sufficient quality magnetic separation. It also removes cobalt from natural waters higher than 25% removal efficiency, and the highest was 71, as I told you before, and also furthermore, isotherm studies show that it's favorable sorption. Maximum absorption capacities are also mentioned, and the highest was 36, and the equilibrium of kinetic studies show that it's single layer hemisorption. So thank you for your attention. If you have question, you can write me a mail or something like that, because we're trying to be a little bit not late. And now I want to ask my colleague, the Galina Luenienė, to present her presentation.
So I will continue Sergei talk on nanocomposites for metal adsorption, and present you some our data on nanocomposites. The aim of this research was to develop environmentally friendly, safe composites to protect water systems, and we synthesize composites based on high-to-zone and graphene oxide, and applied them for adsorption cesium, cobalt, and europium. Various characterization techniques available at our research center were used for characterization. Adsorption of cesium, cobalt, and europium were investigated using batch technique, and based on our results and modeling, adsorption mechanism of cesium, cobalt, and europium was proposed. So I'll start from composites contained mica clay, local origin. It was synthesized use muscovite, graphene oxide, and magnetite and hematite nanoparticles. Characterization was performed using XRD, Nussbauer spectroscopy, and in samples we found nanoparticles of magnetite maghemite of 14-15 nanometers, and spectroscopic analysis indicated various functional groups available for adsorption. So batch sorption experiments was performed. We used solution traced by radioactive isotopes, and samples were measured by gamma spectrometry. The quite high adsorption capacity was obtained. This composite can be used in large pH, in wide pH range, and adsorption isotherms indicated beta agreement with Freundlich model, and adsorption kinetic with pseudo first order model. So now I move to the second eye. Based on this data, You can see here proposed adsorption mechanism. It's quite complicated. It involved electrostatic attraction, ion exchange, inner sphere complexation, cation exchange, and electrostatic interaction on graphene oxide, and also functional groups involved into this adsorption process. So the next next composite synthesized in our laboratory, it's magnetic Prussian blue graphene oxide. The aim of this work was to improve adsorption capacity of magnetic Prussian blue composites and investigate the adsorption parameters and apply them for preconcentration cesium from natural water samples. So magnetic Prussian blue composites were synthesized, and again they were characterized using Bauer spectroscopy, XRD, and TEM. Our results showed that Prussian blue particles were uniformly distributed on the graphene oxide, on graphene oxide sheets, and Prussian blue particles of non-cubic shape were observed in sample number 2-3, while in sample 3.2 very well shaped cubic particles were defined, and particle size was estimated between 5 and 2 nanometers. Magnetic particles also were found in this composite. Their size was about 9-10 nanometers. So FTIR studies and Raman spectroscopy confirmed the different structure of these samples and explain preparation of these samples and effect of graphene oxide oxidation level. We performed the adsorption experiment with cesium, and quite high adsorption efficiency was found for samples 3.2 and lower efficiency for sample 2.3. Removal efficiency reached 90% for sample 3.2 and modeling indicated that the Langmuir isotherm described the adsorption cesium on composite better than Freundlich, indicated uniform distribution of active sites on the surface and monolayer adsorption. Kinetic studies showed pseudo-first we have the second order modeling agreed better with experimental data, and removal efficiency was really very high for these samples at pH 7. Modeling and our experimental data were used to describe adsorption mechanisms, and you can see that adsorption mechanism is quite complicated. It involves ion exchange, ion trapping, and surface complexation on magnetite, and our samples with very good adsorption capacity was used to pre-concentrate cesium from seawater collected at the Baltic Sea, and we have received very good results, and compared our data with standard evaporation method, and you also can see quite good agreement between these two methods. So graphene oxide magnetite hytosand composites were synthesized using slightly different methods. First sample was synthesized using glutaraldehyde, but in two other samples we don't use glutaraldehyde for cross-linking. So samples were characterized using the Nussbauer spectroscopy. X-ray diffraction analysis was performed also, and nanoparticles in samples were identified, and their size varied between 6 and 8.5 nanometers. FTIR spectra indicated functional groups available for adsorption, and Raman spectra again showed very high oxidation level of graphene oxides, and this importance for synthesis of nanocomposites. TEM images showed morphology of these samples. You can see particles on the graphene sheets, sample B, and also small particles of 6 to 8 nanometers can be seen in the samples. Adsorption mechanisms can be described as complexation and electrostatic attraction of europium on the synthesized composite, and also various group of composite involved into this adsorption process. So this synthesized composites also were used for pre-concentration actinides from seawater, and the data on adsorption europium biocomposites from natural water showed efficiency from 70 to 100%, and the pre-concentration europium, plutonium, and americium from seawater also showed high efficiency for all studied composites. So, and as conclusion, I can say that all synthesized composites was of high efficiency, very complicated mechanism adsorption was found, and high adsorption capacity as attributed to the various available groups in the on the surface of composites, and magnetic particles can be used not only for separation, but also they can provide additional adsorption site. And these samples was found to be efficient, non-toxic, and can be used as an environmentally friendly adsorbents to remove metals from water samples. Thank you for listening, and if you can, if you have questions, you can just mail me. Thank you.
Thank you, Galina, for your presentation. It was very interesting. Really a lot of results were presented. Thank you. Now we are having another presentation from Center of Physical Science and Technology of Sonata Pleskite that will show us something about degradation of microplastic.
Okay, so good morning, everyone. Thank you, Sergey, for such a nice introduction. So I am Sonata Pleskite, and I am a PhD student and a junior researcher at the Center for Physical Sciences and Technology in the Department of Environmental Research. And as it was already presented, today I will talk about the photocatalytic degradation of microplastics, and we will focus on the physical LDPE film properties such as size and color, and we will analyze how these properties can influence the photocatalytic degradation. So to start with, polyethylene is one of the most commonly used plastic polymer with an annual global production of 140 million tons. And it's very important to add that polyethylene films are one of the most widely applied plastic polymers in agriculture, especially for the greenhouses and soil mulching. And of course, we need to admit that low-density polyethylene films, they greatly contribute to the modern agriculture. However, LDPE films can be broken down into smaller plastic debris or items, and eventually into microplastics due to the, of course, the environmental degradation and aging processes in the farmlands. And once microplastics are introduced into the environment, it can cause so-called Trojan horse effects, where microplastics can transport the different kind of harmful additives and pollutants, increasing their bioavailability and, of course, toxic effects. And probably here is very important to add that currently there is no such as technically practical or low-cost pathways how we could recycle or reuse soil contaminated microplastics or LDPE films contaminated with soil. Therefore, we chose to work with a photocatalytic degradation due to its high degradation efficiency, also the low environmental impact, and of course, due to its potential to break down microplastics into non-toxic end products. So as a prototype for microplastics, we use commercially available LDPE films, which we bought very simply in a local supermarket. And we found LDPE mulch films in two colors. It was black one and also a transparent color. Also, we, just give me a second. I will use a pointer. Yes, so also we cut these films into two size groups. The first size group was 1 by 1 millimeter, and the second group of size was 3 by 3 millimeters. So to move further, we synthesized silver titanium dioxide nanomaterials by photoreduction method, and these nanomaterials, they contained at different concentrations of silver, and we use them as a catalyst in our degradation system. Also, I want to mention that the presence of a crystalline phase of titanium dioxide and also the successful deposition of silver on titanium dioxide was confirmed by different analytical tools, and the average size of silver nanoparticles was approximately 20 nanometers. And the photocatalytic degradation performance and the efficiency was determined by mass reduction, also FTIR spectral analysis, followed by SEM imaging. And in addition, we also performed the quenching test to determine which reactive oxygen species were the most involved into the titanium dioxide-driven photocatalytic degradation system. So in the figure number 1, you can see FTIR analysis results of the pristine LDPE films with two colors. So the black line represents the black films, and the bright blue color represents transparent LDPE films. And FTIR spectra, they confirm the presence of characteristic absorption bands of LDPE polymers. However, I need to pay your attention that there was slight differences between two LDPE films, even they both were low density polyethylene. So when we are talking about the black films, we can see over there the appearance of a new peak at 873 inverse centimeters, which corresponds to vinyl group. And when we are talking about the transparent films, we can even see more spectral changes in this type of the polymer. So let me start with this region where we can see carbon-hydrogen out-of-plane bending. Also, we found the appearance of new peak formation at 1,661 inverse centimeters, which corresponds to carbon double bond carbon stretching. And then we found that there starts a new formation of a broad band in this region, which kind of suggests or indicates that there is alcohol-containing groups in a pristine LDPE films. And then we also analyzed the surface morphology of both films. So as you can see from figure number 2 in a part A, there is black film, and you can see that the surface of black films were full of irregularities and roughness. Also, we found that there are some flakes like over here and here. And meanwhile, the transparent films, they had quite smooth and flat texture of the surface, and there was no like visible cracks or fractures on the surface at all. So even if both LDPE films were bought from the same supermarket the same day and so on, we already can see that there was some spectral differences between the particles, and of course some surface morphology differences. And these differences, which I just presented, they are going to be very important further when we will be talking about the photocatalytic degradation. But first of all, let me show you the overview of the photocatalytic degradation. So in general, the degradation efficiency, the mass loss of LDPE films increased over the time, and you can see that the highest degradation efficiency was obtained for black colored LDPE films with a size of 1 by 1 millimeter over here, and it reached almost 10% of mass loss. And also I want to add that the highest degradation efficiency was obtained with silver titanium dioxide nanomaterials, which contain the highest concentration of silver. Since our today's topic is to analyze the impact and the influence of physical properties of LDPE on photocatalytic degradation, therefore I will focus further on only on the results which we obtained using silver titanium dioxide nanomaterials with the highest silver concentrations, which are marked in green line over here. And let's start from the comparison between LDPE films with different sizes. We noticed that the higher degradation efficiency was found for smaller LDPE films, which had a size of 1 by 1 millimeter compared to the higher size LDPE films. And of course, very naturally, there is a question: so why smaller LDPE films are more prone for the degradation? And this actually could be attributed to the fact that smaller LDPE films, they have a higher surface area to volume ratio. And this means that higher than larger ones. So this means that a higher surface area to volume ratio maximize the interaction with photocatalyst and also with reactive oxygen species. At the same time, it also enhances the light exposure. And what was interesting at this point, actually, that during the experimental work, we also noticed that smaller particles, smaller LDPE films, they were more evenly distributed in the reaction solution compared to a larger LDPE films. Since larger LDPE films, they were tended to block each other's surface, overlap each other, and of course that reduces the possible active sites for reaction with reactive oxygen species and also catalyst. And the other thing, what we were studying was the comparison between LDPE films with different colors, and we found that black LDPE films with a size of 1 by 1 millimeter had a twice mass loss as it was found for transparent LDPE films. And why color is so important in this study case? So for transparent films, most likely that manufacturers are using strong UV stabilizers, which could be different kind of antioxidants or UV absorbers. And especially the UV absorbers, they are actually reducing and neutralizing the free radicals, which are of course essential for the photocatalytic degradation reactions, like other in case of oxidation processes. And when we are talking about the black films, here manufacturers are using most often black carbon black, especially for plastics which are which we are using for outdoor activities. And why we got the higher degradation efficiency for a black colored LDPE films compared to transparent films could be related to the heat absorption effect, since black films, they absorb more light, and the temperature of the black colored LDPE films of course increases. And so far we of course know that the temperature is one of the key factors when we are talking about the photocatalytic degradation. So the higher temperature we have in our reaction system, in most cases, in most scenarios, the better photocatalytic degradation performance we have. Also, I want to dig a little bit more the surface morphology and the surface changes that we observed. So, of course, the most pronounced surface morphology changes were found in the black films with a size of 1 by 1 millimeter, which is represented in a figure 5 part A. And here you can see the formation of some cavities, like over here, or also you can see more flakes and cracks. And what is probably the most interesting part and the most important, that we also can see the formation of a deep pits. And this kind of surface morphology changes, they are indicating the decomposition of LDPE matrix, which most likely occur due to the volatilization of organic materials and also the main chain scission. So that kind of I would say pretty highly pronounced surface changes are seen in both films with a size of 1 by 1 millimeter and with a size of 3 by 3 millimeters in case of black films. And the part C and D of the figure represents transparent LDPE films. And here we can see that of course there are some changes, because these results are after 16 hours of UV radiation together with a catalyst. But the changes and the effect on the surface of transparent LDPE films, they are not that well pronounced, and we can see that they take a place only on the top layers of our LDPE films, which means that there is no substantial internal damage or defects into the deeper inner layers of LDPE transparent films. And here I would like to remind you that a few slides before I show you the SEM imaging of the pristine LDPE films, and you already noticed that black LDPE films, they had already some defects like flakes and some wrinkles on the surface, on the top surface layer. And we believe that especially these kind of defected regions on the top layers of black LDPE films, they can be the initiation sites for the oxidation processes, and therefore there are more changes obtained after the photocatalytic degradation reactions exactly on the black colored LDPE films. And the following figure represents the FTIR analysis of both color films over the degradation time of 16 hours. So you can see over in the part A, you can see the black LDP films, and in the part B, you can see transparent films. So I will start with a black color, and I'm not going to discuss each of the peak that you can see in the graph, but I want to pay your attention that for black LDP films, after the 1 hour of UV radiation in the presence of catalyst, we already can see the formation of new peak at 1,740 inverse centimeters, which corresponds to carbonyl group. And especially carbonyl group is the main indicator for the successful oxidation process within the polymer chain. And of course, also we can see the formation at 1,654 inverse centimeters, which is attributed to carbon double bond carbon stretching, and this most likely occurs due to the main chain scission of polymer. And of course there are some more branching over here, but I would like to talk a little bit more about the trends and similarities that we found in transparent LDPE films. Also, you can see the formation of carbonyl group over here. You can see the increased peak, which corresponds to the carbon double bond carbon stretching, but probably I want to pay your attention that these changes are less pronounced than in a black colored LDPE films. And in general, to summarizing this slide, I could say that the spectral changes that we found after the photocatalytic degradation, such as new peak formation and the branching of already existing peaks, they are indicating the chain scission, also branching, as well as formation of new methyl and methylene groups, and also they are indicating the decrease in the crystallinity within the polymer chain. And finally, also we evaluated the reactive oxygen species formation, and for this reason we performed the quenching test using reactive oxygen species scavengers, which are already presented in the slide over here. And we found that hydroxyl radicals and superoxide radicals are the main reactive oxygen species responsible for the successful degradation process. And you can see from this graph that hydroxyl radical had the biggest impact, the biggest contribution to the photocatalytic degradation reaction. And this could be related to the high hydroxyl radical redox potential, which is approximately 2 plus 8, sorry, which is approximately plus 2.8 volts. And also what was interesting over here that we noticed that, of course, the composite containing silver and titanium dioxide had a higher photocatalytic activity and performed the better degradation efficiency for black colored and transparent colored, both LDPE films. And probably this is most likely related with silver, which can, in this reaction system, can increase the charge separation, as well as reduce the recombination rate between electrons and holes. So that was also interesting to check it by ourselves. Therefore, in this reaction system, we can see that the contribution of hydroxyl radicals are the highest, but also the contribution of superoxide radical is also very important, which is over here marked in a yellow color. Yeah, so for the summary, I would like to once again mention that in this study, we checked the physical properties impact on the photocatalytic degradation of LDPE films. And for that reason, we used 3 analytical tools. So it was the mass reduction, SEM imaging, and also FTIR analysis. And all of these 3 analytical tools, they confirmed that physical properties such as size and color really can make an influence and differences on photocatalytic degradation. And also we found that black LDPE films with a size of 1 by 1 millimeter were degraded with the highest efficiency, which was approximately 9.8%. And finally, we found that the hydroxyl radical is the primary reactive oxygen species responsible for photocatalytic degradation. And you can see the order according to their impact and influence to the degradation efficiency. And probably the take-home message would be related with a physical properties. So I want to highlight that even minor physical and chemical differences between LDPE films can affect photocatalytic degradation, highlighting the need to optimize reaction conditions based on their properties for the real-world application. And it's also very important to say that these results contribute to development of advanced oxidation processes for the soil contaminated and other kind of agricultural plastic waste. And that would be it. I want to thank you for your attention, and also I am open for questions. And if we do not have a time for questions at the moment, you also can write me an email. Thank you.
Thank you, Sonata, for your detailed presentation, and good luck in your studies. Hope next time you'll be like a doctor presenting another one results. And now I want to ask you to stop the sharing of the screen, and another presentation will be presented by Abdullah Khan that just finished his PhD studies. So we are having the fresh doctor here that will present his results. So Abdullah, please, floor is yours.
Thank you, Sergey, very much. The topic which I'm presenting here today is about evaluating green infrastructure as a natural defense against urban microplastic pollution. So I just want to explain a little bit that why we are studying airborne microplastic and why it is so important. And the reason is that 55% of the urban population is living in urban environment. Global population is living in urban environment, and this will increase to 68% by 2050 according to United Nations. Now, according to World Health Organization, 9 out of 10 people are exposed to bad air quality, which cause 7 million deaths every year. And one of the leading reasons of these deaths is the elevated concentration of particulate matter in the urban environment. Now, particulate matter in urban environment is coming from various sources, but one of the leading sources is traffic, because traffic is not only contributing from exhaust emission, just like black carbon, but also non-exhaust sources like microplastic. Microplastic from traffic-related non-exhaust sources are an emerging pollutant of concern, not because of health implication, but it's contributing 6 million tons of particles in the air, which is almost equal to 10 to 20% to the total load of particulate matter. In the present study, we basically identified 2 research gaps. First one, non-exhaust emission. Microplastic from traffic sources like brake, tire, and road wear are still poorly understood in terms of their composition, abundance, and environmental behavior. And the second one is interaction with the urban green infrastructure. This study aimed to assess the effectiveness of TUYA Occidental SH in trapping airborne microplastic pollution from non-exhaust emission sources, and it's assessing its efficiency at different seasons and also identifying the size, shape, and chemical composition. This study to do the mitigation of microplastic exposure and its viability as a green solution for promoting a healthier environment. The present study was conducted in Kaunas city in the year 2023 and 2024, and a total 22 sample has been collected through passive deposition technique. Here we can see the sampling protocols, that how we collected these sample. So the sample has been collected at horizontal distances, -1 meter near the street, and this is the green infrastructure. So another sample has been collected here, and then 1 meter away, and then 2 meter away. So a total 16 sample has been collected in 4 different seasons at these sampling points. And additionally, 4 sample has been collected in the absence of the H to have a comparative control and experimental environment to understand the role of urban green infrastructure, that how much they are reducing the particle from the air. And additionally, 2 sample has been conducted vertically to understand the particle behavior in the air. After collecting the samples, the sample was collected on glass fiber filter and transferred to glass beaker using distilled water. And then organic matter was eliminated using a 30% hydrogen peroxide solution. And then microplastic are separated using a density separation technique. And then finally, it was filtered through glass fiber and dried at 60 degrees centigrade in an oven for 24 hours. And then the particle size, shape, and color was absorbed using an optical microscope. And the chemical composition was obtained through an micro FDR spectroscopy. Now, here we have the particle number concentration throughout the campaign in 4 different seasons. And here we can see at the different distances. And if you look at the particle number concentration in different seasons, so you will see that we have the lowest particle number concentration in summertime, and the highest particle number concentration has been observed in the springtime. Further, if we compare the experimental environment number concentration and the control environment, so we have seen a significant reduction in the particle number concentration. The number is around 50%. And these particle number concentration variation in different season and different distances is attributed to actually different meteorological condition and the vehicular activities in different season. For example, in the summertime, during the holiday seasons, we have lesser vehicular activities, and that is one of the reasons that we have the lowest particle number concentration in the summertime. Here we have the particle shape, and we can see that in summer, autumn, and winter and spring, we have a very clear dominance of the fragment shaped particle in the study area, and a very less number of fiber has been observed in the study area throughout the study campaign and all distances and in all seasons. And the reason is that most of these particles which is generating in the road infrastructures, they are generating in the fragment shape compared with the fiber shape. Now here we have the color composition in different distances and in different seasons, and clearly we can see that there are different variations of color in the study area, but some of these color, for example, black color, white, and brown color, these are dominant in different seasons, and overall they show a clear dominance in the entire study campaign. And the reason is that most of these black, white, and yellow or brown color, they are coming from the road infrastructure. For example, the fraction between the tire and the road infrastructure. So mostly these particles are coming from tire wear, brake wear, and some of these white and yellow, these are contributing from the lining of the road surface, white lining, which is used for different purposes. For example, these road signs, and some other color has also been observed in the study area, but they are not in very high number compared to this color, but still they show their presence, and it could be contributing from different other sources, from the pedestrian activities or from long range transport in the study area. And now, on the other hand, we have microplastic size, and the size has been classified in different size interval, less than 20 micrometer to microplastic higher than 2000 micrometer particle size range. But actually, two different size range was the most dominant in the study area throughout the study campaign, and that was 50 to 100 micrometer and 100 to 250 micrometer. And the reason for the high abundance of the particle in this size range is that the particle which is generating from the vehicular activities normally in the size range of 100 to 350 micrometer size range, but the most dominant size range is normally between 50 to 250 micrometer size range. Now here we have the chemical composition of the microplastic in study area, and we have 10 different polymers identified in the study area: polyethylene, rubber, polyester, polystyrene, polysulfone, acrylates, polyisoprene, polyamide, polypropylene, and polyvinyl chloride. And we have seen and observed that there's a lot of variations in the chemical composition of microplastic has been observed, not only on different distance from the street, but also in different season, with the most dominant polymer was polyethylene, contributing 43% to the total load of microplastic, and followed by polyester 18%, acrylate 18%, and polyisoprene 19%. And if you look at the chemical composition, so we have these black colors. These are the natural rubbers that is directly as a generation of tire particle, which is contributing around 14%. But polyisoprene is another synthetic rubber component or polymer which is used in the composition of the tire, and that is the reason that we have seen this high concentration of these polymer in this studies area. Now here we have vertical distribution of microplastic inside the hedge, and we have seen a very similar trend. For example, fragment shaped particle is dominant black, followed by brown, and particle in the same size class range, and a very similar composition of polymer has been observed in the vertical distribution as well. But an interesting thing we witness here is that if you look here at the top edge of the hedge, so we have seen that there is an increase of 0.5% of the fiber concentration, and it's indicate that due to the aerodynamic behavior of the fiber shaped particle, that these particles can go into higher elevation and can travel into far different distances depending on the wind speed. A similar trend has also been observed for the chemical for the other polymers as well. And for example, like PVC, polyethylene, acrylates, and polyisoprene. So if you look at these four polymers, so they were completely absent, almost 0% at the middle height, but at the top height, there has been significant results. And it's also an indication that some of these polymer has also, due to its aerodynamic behavior, has the tendency to go into higher elevation and also can travel further as well. Now here we have the removal efficiency of the urban green infrastructure of the microplastic from the air for different shape and for different sizes in different seasons. So here we can see that the highest removal efficiency of the particle has been observed in springtime compared with the other season, and the low in the autumn season. And throughout the study campaign, total 51% of the fragment shaped particle has been removed from the air by the tuya, and 45% of the fiber has been removed by the tuya occidentalis hedge in the entire campaign. Here we have the removal efficiency of the particle in different size ranges, and the highest removal efficiency has been observed for 50 to 100 micrometer size ranges, and the followed by 20 to 50 micrometer size range, which is 53%. And the highest removal efficiency seasonal wise was 94%, and that was observed for 50 to 100 micrometer size range in the springtime. Now here we have the inhalation risk index for different season and risk reduction by Tuya on this side. And we can see that in the summertime, the inhalation risk index and the risk reduction by the Tuya in the autumn and in the winter. And here we have observed that in the springtime, the inhalation risk index was the most highest, 831, and at the same time, the risk reduction was also the highest, 71%. So overall, if we look here, so we can confirm that even the Tuya is not only removing particle from the air in different season, but it also be more effective even in the high pollution episode. For example, like in springer time when the particle concentration was too higher, the Tuya risk reduction was almost the highest, 71%, which proves its potential role in removing particles from the air. Now we have correlation analysis of microplastics, and we have seen that the area closest to the road show a strong positive correlation with small particle less than 20 micrometer and a moderate strong positive correlation with particle size of 20 to 50 micrometer size ranges. A clear inverse relationship has been observed between particle less than 50 micrometer and particle larger than 2000 micrometer, indicating different atmospheric transport behavior and emission sources. Additionally, polyethylene and polypropylene are predominantly associated with larger fragments, while polyamide and polyester are linked to finer fiber, reflecting their different origin from industry and textile sources. A statistical significant accumulation of microplastic here suggests the potential role of Tuya occidentalis H in trapping particles from the air. In conclusion, I can say that fragment-shaped microplastic was the most dominant with 50 to 100 micrometer size range, and color composition and polymer composition revealed that polyethylene was the most abundant polymer across all sample, peaking 69% in springtime, highlighting its widespread presence and persistence in the urban atmosphere. And finally, Tuya occidentalis has significantly reduced airborne non-exhaust microplastic pollution in urban street. The hedge achieved 54% of efficiency throughout all the season, with the highest removal efficiency recorded in summertime for total microplastic 65 and 71% fiber. Furthermore, Tuya occidentalis reduced exposure risk by 71% in springtime when the inhalation risk index was highest at 131, confirming the potential role of urban green infrastructure in mitigating traffic-related airborne microplastic pollution. Thank you very much for listening. So these results are published in the Science of Total Environment. And if you have any question, you can ask now, or if you want to, you can write me an email. Thank you very much.
Thank you, Abdullah, for a very interesting presentation. And now I want to call to present the last one presentation for today that will be done by Aida Pradauskaitė from Nature Research Center, Vilnius, Lithuania. Aida now present us the magnetic hydrogen nanocomposites effect on zebrafish. So floor is yours.
Yes, so thank you for introducing me. I am a PhD student from Nature Research Center, and my colleagues already examined the adsorption capacities of magnetite chitosan nanocomposites, and we did some experiments to evaluate the environmental safety and ecotoxicity of these nanoparticles on zebrafish. So we all know that chitosan is a natural non-toxic polymer with good adsorption capacity for heavy metals and other pollutants. And magnetite, on the other hand, has magnetic properties that attract heavy metals or organic contaminants. And it is also useful as it can be separated from other materials, washed and reused. So what we tested is magnetic chitosan nanoparticles, the MCN, that are a combination of chitosan with magnetite nanoparticles, and they may exhibit even greater potential in adsorption of pollutants from contaminated water. And so yeah, we conducted experiment on our model organism, zebrafish, Dania aurelia embryos, and they were separated into several groups and exposed to different concentrations on contaminants. So this group was a control of water. Then we had the MCN group of lower and higher concentrations, so 20 and 1000 milligrams per liter of MCN respectively. The cobalt exposure group with lower concentration of 0.2 and higher concentration of 20 milligrams per liter, and also the cobalt plus MCN mixture group with lower concentrations of cobalt and MCN, and another one of higher concentrations of cobalt and MCN. And all these groups consisted of 4 replicates, where 5 individuals were observed per replicate. And the parameters that we tested were hatching, heart rate, and behavior, including total move distance, velocity, movement, meander, and turn angle. The MCN nanoparticles were synthesized using cross-linking method, where the agent glutaraldehyde was used to combine chitosan with magnetite in a 2:1 ratio. And so from the very beginning, we could already observe the attachment and accumulation of magnetic chitosan nanoparticles around the embryos of Dania aurelia. So here on top left of the figure, you can see the control group, an embryo that is not affected by nanoparticles. In the middle, you can see lower concentrations of MCN and MCN together with cobalt. And on the bottom, you can see higher concentrations. So those concentrations were really considerable and were visibly noticeable by a microscope. And so what we checked was the hatching rate. Here on the left, you can see lower concentrations of cobalt, MCN, and their mixture. And what we observed is that lower concentrations of cobalt and MCN caused postponed embryo hatching. However, the cobalt plus MCN mixture group seemed to show no difference in hatching rate compared to a control group. And this could suggest us that the mixture has lower toxicity than cobalt or MCN separately. On the right, you can see the image of higher concentrations, and here the hatching rates were similar in all exposure groups. The only statistical difference was in 1000 milligram per liter MCN group that was statistically different from the cobalt group, but not statistically different from the control. We also checked the heart rates. So here on the left, you can see heart rates of Dania aurelia at different time points after exposure, so 24, 48, and 72 hours. And what we noticed is that at low concentrations, MCN and cobalt plus MCN mixture caused a statistically significant increase of the heart rate in Dania aurelia after 24 hours compared to the control. But after 48-hour exposure, only cobalt plus MCN mixture group showed a higher heart rate, while cobalt group had a decreased heart rate compared to the control group. And that after 72 hours of exposure, there were no more statistical differences. We saw similar results using higher concentrations, so the graph on the right. Here, the heart rates were statistically different among groups only after 24 hours exposure period. So all 3 exposure groups, cobalt, MCN, and cobalt plus MCN mixture showed increased heart rates compared to the control group. And as I mentioned, after 48 hours and 72 hours, there were no more statistical differences here. And lastly, we checked the behavior of Dania aurelia zebrafish larvae. Here I put a graph of different behavior parameters, but to sum up, cobalt, MCN, and their mixtures at different concentrations did not have very notable effects on the behavior. There were only 3 parameters that did show statistical differences that you can see here. Namely, only the higher concentrations of cobalt plus MCN mixture group show statistical differences from the control group in Mander, turn angle, and angular velocity of lavi. There were some differences in other groups, however, they were not statistical. And so to conclude, what we observed is that cobalt exposure to Dania embryos resulted in the most postponed hatching rates among all observed groups. Cobalt groups also exhibited changes in heart rate, but did not show statistical differences in behavioral parameters. The MCN seemed to postpone hatching rates, but only at lower concentrations of 20 milligrams per liter. MCN at low and high concentrations caused a statistically significant increase of Dania heart rate after 24 hours of exposure, but not later, and also it did not significantly affect any behavioral parameters as we saw. And finally, the cobalt plus MCN mixture did not show any differences from the control group in embryo hatching. However, cobalt plus MCN mixture groups showed the most significant increase of Dania heart rate compared to the control after 24 and 48-hour exposure, but again, no differences were visible after 72-hour exposure. And finally, high concentration of cobalt plus MCN mixture also resulted in a statistically different of Mander, turn angle, and angular velocity of lavi compared to the control groups. And so to sum up everything, cobalt plus MCN seems to have some effects on Dania, but they do not seem to have acute toxicity. However, to answer more for sure, more experiments should be conducted in this area. Thank you.
Thank you, Ida, for your presentation. And now I think I would give the word for the Reda.
Yes, thank you very much. And I want to thank everybody for a nice presentation. And I wrote for someone to ask name and surnames which participated in this conference, and please send me by mail and we will send certificate of attendance. So it was the last presentation, and the record will be uploaded on Klaipėda University site, and also abstracts of presentation will be uploaded too. So thank you, and it is very nice. It was very nice to hear all presentation which has actual problem on microplastic and nanoparticles in the environment and their effects on human health. So thank you very much and see you for the next time.